And then I'd also like to thank Erica, John, and Andrew for their presentations. We're going to have a panel discussion now, and I'd like to invite, yep, there's Andrew and Erica, great. I just want to remind those who are participating in the webcast that you can use the Q&A box to submit questions. And I'm going to start with a couple questions, and we also have some questions from some of the people participating in the planning group and the drug forum as well. So I'm going to start with a question that, John and Chi and Erica, you all talked about generating data, and Andrew, you talked about using data. So maybe going to put you all on the spot a little bit, and I'm curious on how well those connections are actually working. So I guess Andrew and John, I could ask you on the brain side. Andrew, I know there's some work that twoXAR is doing in glioblastoma, for example, and some other neurological diseases, and so I don't know if you've been able to access or any of the things from the Brain Initiative, or John, if you have examples of where people are using some of the data you're generating in actual drug development processes. So
right. So the Brain Initiative, the Brain Initiative. Go ahead. Is fairly young. The Cell Census Network, for example, started in 2017, and the big tranches of data are just starting to come out. It's been a challenge, quite frankly, in terms of, not in terms of managing it so much, but in terms of finding ways to render the data more accessible to people beyond those who are just generating the data. So some of you may be familiar with the Allen Brain Atlas, which is an atlas of in situ hybridizations in the mouse brain, covering about 20,000 plus genes. If you find a gene, say, in one of your studies, and you're just curious where it's expressed, you just click on the gene name and boom, you get this beautiful set of serial sections showing the localization of that transcript across the entire mouse brain. Now, genomic data are more complex than that. We not only have RNA transcriptomic data, but also epigenomic data, as well as other modalities that are layered on top of that. So right now we're working very hard to make that more accessible. The ultimate, one of the goals is to have this serve as a framework. So for example, if somebody's interested in, say, for example, how microglia are involved in some degenerative process or some aspect of neuroinflammation, they can then take their data from the disease model and look to see what's going on in the so-called normal situation to use that as kind of a baseline. So there are many different ways of looking at that. Another way is we have researchers that are identifying cells and circuits that are involved in specific pathways. And to the extent that we would like to identify new therapeutic targets, if you know the cells that are affected, and if you know the genes and proteins that are expressed in those cells, then you have kind of a good starting point. And what we're hoping to do is that if somebody can point to using limited data, say, well, it's this cell type, and find the data from the larger repositories that gives a more full accounting of the properties of those cells, that might be a very quick way to get to the point of having information that's actionable. So we're on the upswing, and I hope that we'll see more general use of the data as we go along. But maybe Andrew can comment on how he and colleagues might find those kinds of data useful.
Yeah, well, I think what you described very well illustrates sort of the data landscape of biomedical data that's available. I like to think of data sources as just a puzzle piece. And so if I'm trying to understand underlying pathogenesis in a disease area, therefore to come up with a new target, it's really about collecting puzzle pieces, and I want them to be statistically independent. I want them to give me different views that I can use to ultimately, create those biological models. And so, what John represents, I think, as a data source is pretty typical from all the sources we work with, and we work with many. They all have some special thing that they measure, and they've shined their flashlight into some area of biology. And for someone like me or our company who's a consumer of these data sources, it's really about building a very strong network of relationships with people that do generate data, really understanding and being able to quantify like what is the if you will, the area of information that data represents, and then working to figure out how we can, really from an economic and from a relationship standpoint, basically get enough data or enough diversity of data together where we can actually make a good prediction. And that, today has a lot to do with building relationships with people and understanding those data sources. I think one of the other speakers was talking about, the desire to have centralized repositories or just more formal ways for people to share data. The bulk of our data comes from, we get into a disease area, we meet KOLs, we meet, research experts in that area, we build new relationships, and eventually we sort of uncover those very sometimes hidden data sources. And that's how we build data models. But that's ultimately how it works from a process standpoint.
Chi, maybe you want to comment on, you talked about sort of some of the pre-competitive consortiums for data sharing.
Sure, thank you, Howie. I really like what John and Andrew mentioned about collecting the puzzle pieces about data sharing. I think data sharing and pre-competitive collaboration is really key to the success. I think despite the vast amount of data generated and availability of really powerful new analytical tools, I think the scientific breakthroughs in most prevalent chronic diseases is still limited, except in oncology. And personally, I think data silo is the rate limiting step. To really unlock the power of the advanced analytical tools such as AI and machine learning, especially deep learning, we need appropriately large, diverse, and high quality data. And data sharing and collaboration are very, very critical, and we need more consortia and public-private partnerships. I think actually in oncology, this is one of areas that is done relatively well. For example, the NCI's Genomic Data Commons is a really good example where the community can work together to promote precision medicine in oncology, but we need much more this kind of collaborations.
Okay, thanks. And then Erica, we obviously are focused on chronic diseases, and as she just mentioned, cancer has maybe done a good job of this. So I know research you are doing is in the cancer area as well as cardiovascular, and I just wanted to ask you, and others can join in afterwards, if you see any differences in the work you're doing or the approaches for cancer versus, we'll use cardiovascular if you want to as a more traditional chronic disease.
Yeah, I think one thing, I was listening to the conversation about data sharing, and I think data sharing is challenging for populations who are underserved. Sort of this idea of broad data sharing can be very uncomfortable for underserved communities, particularly communities that are maybe smaller. So if you know something about the genetics of a few people in that community, you're going to know something about the whole community. So there's concerns about stigmatization and sort of privacy concerns about that. So I think what I highlighted in my talk is a little bit about how can we think about being flexible with those data sharing models, because I think if we just say everyone who participates in clinical research needs to have open data sharing, it's going to turn a lot of populations off, and there'll be a lot of communities that will just say, well, okay, it's not worth it. It's not worth the risk for us to participate in this, and that will only grow the healthcare and research disparities. So I just wanted to kind of make that comment about data sharing, that there are some caveats, and it's important to engage with communities to discuss where they would like data sharing to be and what they feel comfortable with and what they don't. As far as some of the differences in the cancer work that we do, I think our partnership started in the area of cancer because is a real interest of the community. And we have found some unique genetic variation within the community I work with for some of the cytochrome P450 genes that are involved in drug metabolism. And, we wouldn't have known that going in. We wouldn't have, we predicted that we would see differences, but we didn't know what they would be. So it's really important that those studies are done so that we know what the variation are in specific populations, and then therefore we can apply pharmacogenetic testing more equitably.
That's great. And thanks for reminding us about the issues, because there's such a drive for diversity. So coming up with the ways of making the data available in an anonymous way, or maybe it's pooling data from many diverse sources so that the data is available for those who are using it, but it's not directly, linked back to, a small source of data. Maybe that would work. That's great. So a couple questions from one question, actually, if we can, is it Dr. Romero had a question? If they want to unmute and ask that directly to Chi.
Yeah, thanks, Kui. The question is essentially for you. If you could comment on the differences in terms of evidence generation in general, but more specifically signal processing from digital health technologies when you're considering them, on the one hand, as medical devices versus, on the other hand, considering them as drug development tools.
Thank you. That's a really great question. Like there has been a lot of exciting advancement in recent years for both medical device and for digital health tools, either in the medical device side or as tools for drug development. There's a lot of similar considerations with those when those digital tools are used for as medical device or as tools in drug development. For example, both of them will need to consider analytical validations and clinical validations, but there are also some differences. A device that is cleared by CDRH as a medical device does not necessarily mean it can be used for like tools for drug development. And there are multiple considerations. Beyond the performance of the tools, there's also the question of the association between the markers from the digital health tools versus the clinical endpoint that is usually assessed in the clinical trials. And also, what is the magnitude, for example, for the so-called digital biomarker collected from the digital health tools as compared to the clinical endpoint that we are usually evaluating. So the consideration is there's differences in medical devices and devices used or biomarkers or endpoints used in clinical trials. And the clearance as a device does not guarantee the usefulness in clinical trials and vice versa. So that's a really good question. Thank you.
Thanks, Pete. Great answer.
Great. And before we go on to questions from the audience, just want to come back to the notion of the chronic diseases versus more acute diseases. So, Carl has pointed out the benefit of the rapid data sharing that helped with identifying monoclonal antibodies for, against the coronavirus. We've had, lots of cancer examples. So maybe I'll start again with John and Andrew. John, since you're looking across, cancers presumably of the brain as well as, more chronic neurological diseases. And Andrew, you have, cancer things in your pipeline as well as, lupus and the example you gave us as well as other things. Is data just sort of data, or for some diseases are they, I guess I think somewhat of chronic diseases maybe being more complex because they're more multifactorial, but is it just data is data, or is there something different about the acute diseases versus the chronic ones that we should think about and try and act on?
Yeah, look, I think my perspective on it is that in general, I mean, I guess I'm not talking about the pharmaceutical industry, which is very different than just research as a whole. But, my take on pharmaceutical industry is the industry is always going after the lowest hanging fruit, right? Like there's R&D dollars to spend. There's some risk as to whether or not you're actually going to get a product on market to, justify that spend. And therefore, based on your expertise or based on, what else has already been done, what's the thing that we can pursue with our, whether it's modality or technology or process that's going to, get us to that next, low hanging piece of fruit. And so because of that, unfortunately, if you've got a chronic disease that, kind of doesn't really kill you and doesn't really make a massive, difference in your life, but it's just a painful and uncomfortable thing to deal with, it's much harder, I think, especially if you've got an underlying sort of biology picture that's very complex. It's a disease where, again, that underlying pathogenesis is not well understood. Maybe you've got some complex system biology going on. This is why typically metabolic and immune inflammatory fall into this. Just the sort of energy, if you want to call it, but the R&D energy required to go make a breakthrough in that area is just so high. And so I think it's a really good topic to address, because then the question is like, okay, like what are ways that we can address these diseases? And I think sort of, as we've obviously been talking about computational methods and things like that, and hopefully it came out in my presentation, like it's just so darn efficient, where diseases like lupus in the past, energy certainly has been put into seeing if we can't get better therapeutics. But now we can for just far less resources come up with some new and potentially clever ideas that allows us to potentially unlock diseases of that class, right? And so that's a little bit of how I see it. I mean, each disease is sort of its own little mystery, and it's got its own attributes around it. But at the high level, that's certainly how I think about it.
Yeah, just to echo some of that perspective, certainly having those disease-specific sets of information will be critical in terms of lowering the activation barrier toward getting to therapeutics. Just to clarify a bit, the Brain Initiative is the mission of the Brain Initiative is to develop tools in a disease-agnostic way. It's not to say that we're not interested in enabling disease research, but what we're hoping to provide is a framework upon which we can hang the disease-specific projects. So to the extent we can provide tools for interrogation, tools for modulation, in this context, data resource tools that can serve as some basis for a comparison, I think that will be highly enabling. Now, I think it's almost a truism that the success in finding a therapeutic, a drug, goes up markedly if there's genetic validation for that target. And so what we're hoping to do is provide the tools that will allow faster validation, whether it be genetic or otherwise, that give confidence that the researchers are going after the right target. Another thing that came out of our cell census project, which was quite interesting, is we performed an analysis of brain cell types and primary motor cortex across 3 different mammalian species: mouse, non-human primates, and human primates. And what we found is that, depending on your point of view, not too unsurprisingly, the vast majority of cell types are conserved across mammalian species. I mean, this is kind of how nature works. But we also found that there were some notable exceptions. There were some specializations in the primate lineage that arose there and were not evident in the rodent lineage. And moreover, even cell types that were thought to be in common between the two express different sets of marker genes. So when we hear about the issues about taking a discovery, say, in one model system and translating them successfully or unsuccessfully to humans, I think these kinds of data can help guide us toward or away cell types or molecules that, if they are shared in common between the model system and humans, then that's a better bet than if they're not. So I think there's a lot of ways that we can use the data in an intentional and hopefully a more guided way to enhance the chances of success to go from basic discovery to an actual clinical application.
Yeah, and I think just to put a point on that, I think what's really exciting about artificial intelligence is John's talking a bit about different ways to get signal without any discussion on like, is it relevant to a disease area? And like that to me is super exciting. And the reason is what artificial intelligence does is, unlike a human who's got sort of a thought process behind, relevancy, the machine doesn't care. It's just looking for these, if you will, statistical relationships, like what's a confounder and what's actually, really contributing to disease pathology, and can pick those things out. And so as a data scientist and someone who's exploring this information, we get super excited when we see, we call it statistically independent data, but basically data sources that are measuring something that other data sources aren't, because that's this rich area to find new puzzle pieces, to find new pieces. And I really enjoy, researchers who are like, look, I'm just going to generate data, with no disease hypothesis, but rather just to see like, where can I find these differentiated signals? And that's the kind of stuff that AI just feeds on.
It's great. Good insights. So a couple questions from our audience. And the first leverages some of the things we heard this morning, which was about building sort of biorepositories and how that might feed into discovery and trying chronic disease specifically. So I know we've been talking just now about data sources and things, but this would be looking a little bit more at the biorepositories. And so maybe one way to think about this, if you want to start, Eric, is setting aside the data sharing issues. If your dreams could come true about how the information you're collecting could lead to a breakthrough in a chronic disease for the populations you're studying, how might that work or be helpful?
Yeah, thank you. I think all of us want to see the drug discoveries and developments that are coming out be broadly accessible for all. And, we can't just kind of keep doing the same studies and recruiting the same types of people into the research and using the same models and expecting nothing will change. So I think that, having a very deliberate community-based way to engage communities to say, hey, is this research you're interested in, and how can we increase and diversify those biorepositories? As far as my long-term vision, I partnered with the Confederated Salish and Kootenai tribes. They partnered with me, I should say, rather, such that they were really interested in being able to utilize pharmacogenetic testing in their clinic. So, the concerns from the physicians there were that available tests might not be useful for them because they're based on findings and genetic variants known to be causal in European populations. So there was concerns that when they are ready to implement that, it might not be useful for their patients. So that was really the genesis, was that, can we provide more data to help aid development and implementation of pharmacogenetic testing into the clinical workflows for tribal health. And that's our long-term goal.
Great. And is there, I know you're, as you described, you're working with, some of the local tribes. Is there any, so at the risk of generalizing, is there research being done with some of the other tribes to see how much variation there is across, the Native American population, or are there some similarities?
There is not actually a lot of data in indigenous populations in the U.S. Our work, we first published in 2013, and there has not been a lot more work since then. I do have some collaborators working with with Alaska Native communities in Alaska. So we're part of a larger research network that includes researchers in Alaska, Montana, Oregon, and Washington that have there are several community-based partners. So we do have some data to compare in Montana versus Alaska. There is not, honestly, too much other work in the United States. I know there are efforts to do more genetic research in other indigenous parts around the world, in the US. I can say that there are quite a lot of differences between the genetic variation we've observed in Alaska and in Montana, and also not just genetics, but dietary predictors that can influence chronic diseases, that can influence response to medications for chronic diseases. And yeah, we see quite a lot of differences between indigenous people in Montana and Alaska, which makes sense. The food sources are very different. They're quite far away from each other geographically. So yeah, I would not expect that what we can find in Montana will be completely generalizable to other indigenous populations. And I think it's really imperative that we have more researchers out there working with communities so that we have a better idea of the relevant genetic variation.
Okay, so that's a good insight, is that in terms of building these biorepositories, they need to be broad as well as deep, right? That's yeah. Great. Yeah. And then another question I'm going to focus in on, again, people are interested, obviously, in how we can use some of the techniques broadly across chronic diseases, and we've had some good discussion around that. But this morning, our actually kickoff speaker talked about Alzheimer's disease, and so just any thoughts about, is Alzheimer's just another on the list? Andrew, you might just say, well, it's just another disease on the list, and away I go. Or John, I know you're obviously generating, tools and things that would be helpful for Alzheimer's disease. And, Chi, maybe you've also seen some things here, but just any thoughts about Alzheimer's and specifically, and how some of these tools might be applied to understanding that.
Yeah, I would say this is maybe a little less technology, more just the reality of creating therapeutics. Different disease profiles have very different, if you will, tolerance profiles. So look, if you're, in an oncology situation and you're expecting to die in the next 3 months, you will be happy to take, a therapeutic that's going to have some pretty nasty side effects if it saves your life. Obviously, in the neurodegenerative space, if you've got a disease that, slowly is going to progress over the course of decades, and, we know this, patients have the choice of, feeling nauseous on the couch, and maybe, having a better health outcome, or just, not taking the drug, like the patient chooses to not take the drug. So I think not only is there this biological complexity to the disease itself and the, lack of, I think, full understanding of the disease biology, to therefore come up with a therapeutic. Now you've got the added layer of, should I get this disease one day, and I'm imagining the conversation with my clinician, well, this is going to progress over the next 20 years. And so, here's a medication. Like if the result is, I can't tell I'm taking the med, happy to take it, right? If who knows what those side effects are, but I'm going to be constantly thirsty every day for the rest of my life and get, So I think those challenges really kind of seep into some of those disease areas. And that now it becomes sort of this exponentially more difficult task to not only come up with something that is efficacious, but have something that has that minimum tox profile. And that's really hard to do, for sure.
And Howie, I want to make a comment here. I know we have already talked a lot about data sharing and pre-competitive collaboration, but talking about Alzheimer's disease, that just reminded me of some very exciting collaborations in this field. Critical Path Initiative has taken the lead in a consortium called, I think it's called Coalition Against Major Diseases. And one of their research project is on Alzheimer's disease. And I know there's a lot of collaboration to work on, for example, disease modeling for Alzheimer's disease, etc., which can be very beneficial for drug development in this field. Thank you.
Yeah, if I can just add to that, Andrew, I think you bring up a very important contrast between, say, a patient at end-stage cancer versus some of these neurodegenerative diseases. The timeline and tolerance for side effects is definitely going to be lower. Alzheimer's is certainly, I think, it's fair to say, a public health crisis that we're facing. And I think one of the biggest challenges that whereas we know a lot about what happens at the end stages of the disease, right? When plaques and tangles are manifested, where cognitive decline is clearly an issue, we really know very much less, in fact, very little about the initiation of the disease and how it progresses in the early stages. And I think the key here will be to understand these early mechanisms and really to find ways of preventing progression of disease, even pre-symptomatically. And this is where you get into a real bind, because giving an otherwise healthy person a drug for the rest of their life is, you really have to make sure that your A, it's indicated for that individual, and B, that it will be efficacious with minimal side effects. And again, this is where it's really critical to understand the basic biological mechanisms of disease initiation and early stages of progression. And again, while we're not focusing per se in the Brain Initiative on these specific disease models, they will intersect with what we're doing in terms of looking at so-called normal healthy brains. We just had this really very informative workshop on tissue standards and tissue procurement for a human brain cell atlas. And it was pointed out quite cogently that, well, what's a normal human brain? If you look at 100 people, you'll have 100 different brains with 100 different characteristics. So I think we're going to need to be able to make sense of the so-called normal data to make sure it is representative of the diversity of humans that are in our population, and then to find ways of leveraging that information for the disease-specific research. And in fact, there are some very robust, recently funded studies using some of the very same techniques as being used in the Brain Initiative for studying patient samples from Alzheimer's populations, and it'll be very, very important to closely align these studies so we can actually get the most out of the data as they start coming on board. And it's not just Alzheimer's, but other degenerative diseases. It'll be very important, again, to identify the basic mechanisms that are involved early in disease initiation and progression.
That's great. So one, two more things I'd like to ask about. One is another thing we heard, and I'll use Alzheimer's as an example. We heard this morning about how there were questionnaires that were put together for patients. One of the quote unquote diagnostic techniques, or few diagnostic techniques for Alzheimer's, is patient questionnaires, and they were put together a long time ago. And a little bit to your point, Andrew, they were asking patients about things that weren't really important to the patients. And so more recently, groups that are working on this have actually started asking the patients what are important to them. So any thoughts about bringing in that community part into the process? So in that example, asking what's important. Another one in the neurological area that I've had students in the past who've been interested in is analysis of speech, and the research that shows you can start to see maybe Parkinson's and some of the other neurological diseases, based on speech pattern. So the general question there is about, I'll call it interaction with community, but it's maybe engaging patients more in the research and discovery process. And Erica, you're obviously at some level doing this as well. So anyone wants to comment on that?
Maybe I'll take a first shot.
Yeah.
I think
sure, that'd be great.
You raised a really, really important point. I think to really hear about the voice from the patients, engage the patients to understand what is important to them is really important in drug development. And it's especially very important to the regulatory agencies. FDA has been working on patient-centered drug development. And what you've mentioned is one of our key missions. And also not just FDA, the European agency is also working on how to incorporate patients' voices, engage patients into the regulatory process. Thank you for raising this point.
Yeah, I'd like to add that I think patient and community engagement is really essential, and maybe working to develop multiple tools for different groups. What would be most valuable for them to communicate to their constituencies to increase participation in research and management of chronic diseases. And I think we don't have to look too much further than the delivery of the COVID vaccine now that there is different levels of hesitancy in different racial and ethnic groups around the country, and targeting that message to different communities will be really essential for the uptake of vaccines as well as other health developments.
This is certainly a very important point. I'm glad it's been raised, and I really appreciate these comments. Building trust, of course, is key. I'm starting to appreciate, at least when we think about the patients involved in these studies, the human subjects, as it were, perhaps that's the wrong way to think about the folks that are involved in it, not as human subjects, but as research partners. I mean, they're putting the humans that we involved in these studies are not only our partners, but as are their families. And so we need to really approach that with a large measure of respect to build the trust, especially among communities that have been underserved or actually not well served by the biomedical community. If we are to hope to really have a greater impact across the entire population.
That's a really great point, John, is thinking of research participants as partners in the research and not subjects. Yeah.
It's one of those subtle, seems like a subtle switch, but can make a big difference. So the last topic I'll ask about is our next session next Monday is we focused on the discovery and early stage here. Next week, we're going to transition to later stages. And so I think Andrew and Chi, you both mentioned, for example, how AI can be used across the spectrum, but just Any thoughts? Any thoughts about later stage development and whether it's some of the brain initiative tools, or Erica, some of the research you're doing, or Chi or Andrew, the things you're doing, and thoughts about later stage development for chronic diseases?
Well, I can certainly make the general comment. I mean, our focus is obviously in the very early stages of drug discovery and development. But, part of the reason I opened the talk with just sort of a general view of like what artificial intelligence does and what you can use it for is, again, sort of answering these questions under uncertainty. And so, there's many years ago, I was in a class at Stanford, and we were learning about, all the aspects of, what goes into, making great therapeutics. And at the end of the series of lectures, the professor asked, okay, so now that we've learned all this, what's the number one thing, that matters? And of course, the entire audience, of students got it wrong, because the answer was simply patient compliance, right? Actually taking your medication. And, I think kind of to the previous, discussion here, thinking a lot about, I think, patients as humans and how they make decisions. I think it's, nothing is more frustrating to hear, we've got, all sorts of trouble with growing prevalence in type 2 diabetes. And I hear folks will say, well, just, diet and exercise, why is it so hard? And yet, someone puts a piece of blueberry pie in front of me, like it's really hard to say no. And sort of there's just this, I think, reality of the human condition and how we behave as humans. And I think there's a lot of things that we can do outside of just, the hardcore research itself. And in my outside of my work, I do a lot of things with the homeless community, and just very simple things like just giving someone a home and giving them all the facilities they need, rather than trying to, supply them with emergency services, like completely turns around people's lives. And so that might be a little off topic for what we're talking about today, but the point of what I'm trying to share is, I think sometimes we get focused in the technology or the research or the very specific domain thing we're going after. And if you kind of step back and look at it from, we want to improve, the human condition and unmet medical need on the whole, like what are the best ways to do that? And I think that, part of it is science, but part of it is social. There's lots of different, I think, avenues to explore.
Maybe I can go next.
Yeah, go ahead, Chi.
Thank you. I think most of the topics we talked today are relevant for not only drug discovery, but also drug development stage. And since Andrew already covered AI, so I think I will probably cover another topic. What I want to talk about is modeling and simulation. I think modeling and simulation can be very powerful tools to improve the efficiency from the transition from preclinical to the clinical stage. They can be used to integrate information from different sources to predict the drug effect in vivo and even to predict trial results. In my presentation, I talked about MIDD, model-informed drug development. And other modeling approach mentioned there are both applicable to drug discovery and drug development. For example, the QSR model can be used to predict biological activity of compounds based on their chemical structures. And of course, like QSR right now are even more powerful when the AI and machine learning algorithms are used. And also the PPPK model or the interspecies scaling models can be used to predict the PK of the drug in humans. And sometimes even preliminary PK/PD simulations can be done before the clinical trial are conducted. This in silico modeling and simulation approach can be used to predict the probability of success of the compounds, de-risk the drug development program, and also help with the clinical trial design, for example, selection of the dose to be studied. So I think this kind of modeling simulation approach can be used more often in both discovery and development stage. Thank you.
Yeah,
Eric or John? Yeah, go ahead, Eric.
I would like to add too, I think sometimes it's easy for researchers to get a little bit myopic to say, we're working on this one thing, but ultimately if our goal is to make an impact in the treatment of chronic diseases, we need to be talking with clinicians too about how this can be used in their practice, how this can be used in their healthcare system. So keeping in mind that implementation side and how do we move this research into actual benefits for patients.
John, anything you'd like to add?
You know, a lot of great points here. Our point of view is to create a larger and more effective data ecosystem that is accessible to more researchers, but also will, as Erica mentioned, be taken up by the folks delivering the care. And I think it's going to be a two-way street as well. We have a lot to learn from the people taking care of patients and vice versa. So again, having a more seamless data ecosystem that makes these resources available for more general use will be, I think, a good way to go in the future, or to continue growing toward in the future.
That's great. Yeah, these are great comments. And it makes me think about when I've looked in the past at understanding a disease area and the treatment of that disease area. There's a framework I've used where you typically, as you've pointed out, there tends to be a focus on the doctor and the patient and the drug or the therapy. And the framework that I've used is really starting at the very beginning is where do the patients come from? How do they find out about, the treatment options and things, all the way through, the compliance and refilling their prescriptions or going back for their follow-up treatments. And that taking that more expanded look really is helpful in terms of making sure you ultimately come up with, a therapy that's going to help people and that they're going to embrace. So I want to thank all of you for your great comments and presentations. I know I've really enjoyed the discussion and I've learned a lot. This has been great because we've really seen how the biology is advancing and the ability to generate data is really advancing, and then the techniques to actually take that data and learn way more from it than has been possible in the past is really, really exciting. And clearly there seem like there are opportunities, things being done in chronic diseases and opportunities to do even more. So I'm going to turn it over to Anantha, who I think is going to sort of sum things up and probably give us a little preview of the next session next week. So thank you again, Chi and Andrew and Erica and John.